single nucleotide change, SNV, or when they are common, SNP, polymorphisms. And these variants arose from ancestral mutational events, and when they have a large impact on the individual, then they cause monogenic diseases. But many variants have more modest or minor effect, and they contribute to common disorders. And these variants that contribute to common disorders can be common or rare. And for common variants, we now have pretty much a complete catalog of them across the world population. And these have been obtained by large-scale projects such as the HapMap and the 1000 Genome. And the outcome of that is that we now can genotype them very efficiently using SNP arrays. So our latest SNP arrays can genotype more than 1 million SNPs at the same time. Whereas when we study rare variants, because these are so numerous that even despite our large studies and databases such as EXACT and GNOMAD, we still have many that are undocumented. And that's why in the study of rare disorders, rare variants, we need high-throughput sequencing technologies. Of course, that has progressed and become cheaper, but SNP genotyping technology is still an order of magnitude cheaper than high-throughput sequencing. And that is why, for the study of common disorders, genotyping is still more popular than high-throughput sequencing. So these technological developments have enabled association studies to be done on a whole genome scale. So comparing the frequencies of variants in patients and controls to find variants which may be high risk or protective for disorders. And because it involves testing so many SNPs, there is a risk of false positives. And in order to control the number of false positives to a low level, we needed to adopt highly stringent p-values. The standard is 5 times 10 to the minus 8. And what this means is that we need large samples in order to reach those significant results. And an example of a very large genome-wide association is for schizophrenia. So this study was published in 2014 and combined data from, I think, about 50 studies with a total of 35,000 patients and I think almost 50,000 controls. Sort of this huge international collaborative effort was able to detect 108 loci for schizophrenia, which is a complex, severe mental disorder, and sometimes referred to as the graveyard for geneticists in the past, because it was thought to be so complex and so hopeless. But we now have 108 independent loci and more coming as well. And these sort of outcomes also apply to other common disorders. So this is a graph taken from a paper in 2017 summarizing the status of genome-wide association studies at that time. You can see how the number of discoveries of associations have increased over time and reaching over 10,000 significant associations by the end of 2016, and this has grown even further in the last couple of years to almost 80,000 robust associations for over 300 common diseases. And among these associations are genes that we already are targeting in our treatment of common disorders. The two prime examples are firstly for schizophrenia, because our drugs that we use to treat psychosis are all working on the dopamine D2 receptor. They all are antagonists to dopamine D2. And one of the 108 associated loci was DRD2. And another example is the treatment of hyperlipidemia. So statin can lower blood lipids, and one of the genetic associations for blood lipid level was the gene HMGCR, which controls lipid levels. And one of the observations from that is that the effect size of the natural genetic variant was rather small compared to the effect size of statin. And so a small effect size from the genetic study does not necessarily mean that a gene is not an appropriate target, because the natural variant may have a relatively small effect on gene expression, whereas if we were to deliberately design a treatment, we could design something that has a bigger effect than the natural variant. And this observation that genetic findings may predict drug targets has been taken up by drug companies. So a few years ago, we had a collaboration with GSK, and they looked at their drug development pipelines, and we observed that the drug targets in these pipelines at a more advanced stage tend to have more support from genetic evidence compared to targets that are earlier on in the drug pipeline, the preclinical stage. And this suggests that having genetic support increases the success of drug development. And the list of genes produced, when we have tens or hundreds of genes, can give us insights about the biological processes and pathways that are relevant for a disease. And for psychiatric disorders, an analysis of these genome-wide significant variants have implicated the synapse and the immune system and histone methylation as important pathways for mental disorders. And in our own study on Han Chinese GWAS schizophrenia, we also identified association with markers on the long arm of chromosome X, a location that contains a MACP2 gene, which controls DNA methylation. So again, sort of indicating the relevance of the importance of gene expression in mental disorders. And so we were quite pleased that the study on copy number variants in schizophrenia also identified chromosome X Q28. And so we have a convergence of common variants detected by GWAS and rare variants, these copy number variants, onto the same genomic locations in the same genes. And the CNV analysis on these 41,000 subjects also implicated genes that were implicated in the GWAS, so namely genes that are important in the function of synapse, genes that are relevant for glutamatergic neurotransmission and dystrophin and interacting partners. And in our studies, we also found associations that are specific to a population. Our study on blood lipids on Asian populations, 47,000 East Asians, including subjects from Hong Kong, reviewed 14 variants that were not found in other populations, either because these variants are rare in other populations or they have a smaller effect in these other populations. And interestingly, when lipid levels were studied, the protective variant for blood lipids, variants that associated with lower blood lipid levels, may be the high-risk variant for another disorder. So this gene, the TM6SF2, is a gene that has a variant that lowers blood lipid levels, but the same variant increases the risk of fatty liver disease. And the mechanism appears to be that this gene is responsible for the transport of lipids from the liver into the bloodstream, so that when the activity of this protein is blocked, is lowered by mutation, then the reduction of excretion of lipids into the blood results in lower blood lipid levels, but then the lipids accumulate in the liver, causing fatty liver disease. So I think this illustrates the importance of understanding the full spectrum of an effect of a mutation before it can be targeted for treatment. And just as a single variant can affect multiple traits, some diseases, different disorders, can be influenced by the same genes. And we demonstrated this using twin studies for schizophrenia and bipolar affective disorder. So this was done almost 20 years ago and recently confirmed by GWAS. So there was significant overlap between the association for schizophrenia and bipolar affective disorder. And this has been extended to a range of other disorders. So you can see here that psychiatric disorders tend to be genetically correlated, and also metabolic disorders, different metabolic disorders, also tend to be genetically correlated. And so this genetic sharing between disorders is greatest for diseases that affect probably the same cell type or organs or system. And our genetic findings from common disorders have also been helpful to epidemiologists who have tried to uncover the causal relationship between risk factors and diseases. And this is because one of the problems for epidemiologists is confounding, that even when a risk factor and the disease is associated, it could be because of some confounder. And one solution is to use an instrumental variable which can only affect the disease outcome through the risk factor. And genetic variants provide possibly very ideal instrumental variables, and not only one, but multiple, so that when multiple genetic variants, when used in these Mendelian randomization studies, provide a similar, consistent estimate of the effect between the risk factor, the exposure, and the outcome, then this will give much more confident results, as in this case, for non-high-density cholesterol and heart disease. So the results from both common variants and rare variants, and also our own study, are highly consistent, so indicating that non-high-density cholesterol is indeed causally related to heart disease. And for polygenic disease, every person carries a certain number of high-risk genetic variants, so it's just a question of number. You know, we all carry some high-risk variants for every disorder, and we can now accumulate the effects of all these high-risk variants present in a person to give a polygenic score, and a population can be stratified by polygenic scores into deciles in ascending genetic risk. And for schizophrenia, now we have enough data so that the highest and the lowest decile in terms of the polygenic score now differs substantially in risk, about 8 to 20%, as shown in this graph here. And we can also construct polygenic scores for risk factors such as lipids, so not necessarily directly on the outcome, but also for risk factors, shown here. And it can be applied, say, to cancer screening, because someone with a high polygenic score may reach the risk level which are recommended for the commencement of screening at an earlier age compared to someone whose polygenic risk score is lower. So perhaps needing to have regular monitoring maybe somewhat later, and the polygenic score can be combined also with family history. And recently, We also looked at whether genetic variants predict drug response in relation to schizophrenia. So we identified subjects who responded well and responded poorly to antipsychotic drugs, and we find that there are 2 gene sets which contain an excessive number of rare damaging variants in the poor responders, and these 2 gene sets were associated with reduced NMDA-mediated synaptic currents or reduced AMPA-mediated synaptic currents. And of course, these mechanisms are important in memory and learning, which are important for schizophrenia. And current antipsychotic drugs target dopamine, so perhaps it is not surprising that patients with genetic mutation in another system may not respond so well. Okay, I just want to finish with what I think are some of the future trends. So I think we're going to see increasingly large population cohorts which have very comprehensive phenotypic assessments, and assessment of lifestyle, environment, and so on, so that we can sort of look at all these genetic and other factors jointly on a large number of outcomes. I think whole genome sequencing is going to become more popularly used, probably replacing SNP genotyping eventually in a few years' time, and linking up with other data domains, such as clinical data, will be very powerful, and there will be increasing integration with genome functional annotations, such as the ENCODE, epigenome roadmap, and so on, and I think probably greater use of genome functional assays to evaluate the consequences of genetic mutations. So thank you very much for your attention.
Thank you very much, Pak. Our last speaker for this session is Professor